Usually when we talk about a medical diagnosis, there's this underlying expectation of precision. It almost feels like engineering.
Right, like it's a math problem.
Exactly. You fall off a ladder, you break your arm, the x-ray shows that jagged white line, and the doctor just points at it. Broken or not broken? It's a very clean, visible, categorized reality. Welcome, Trailblazers, to the Digital Pathology Podcast, by the way. I'm so glad you're joining us for this.
It is comforting to have that kind of binary certainty. A physical break has a physical-mechanical solution.
Yeah, but then you step into the world of oncology, specifically the task of predicting how a patient's cancer is going to progress after that initial terrifying diagnosis. And suddenly that metaphorical x-ray machine is just broken. We're looking at a diagnostic landscape that is incredibly murky.
Oh, absolutely. And nowhere is this more apparent than in cervical cancer. It remains the fourth most prevalent cancer among women worldwide.
Which is wild to think about, given the vaccines and everything.
Right. We have made tremendous public health strides with HPV vaccines and widespread screening protocols. But once a patient is actually diagnosed, the 5-year overall survival rate, it still hovers around 66% globally.
And the really tricky part for oncologists is figuring out an individual patient's specific survival timeline. Because, we have tools like the FIGO staging system.
The FIGO standard.
Right, which categorizes the cancer based on how far it's physically spread in the pelvis. But predicting a specific patient's overall survival, their OS, is still profoundly difficult. And for the trailblazers listening, you know exactly why this matters. Getting this prediction wrong has just devastating consequences.
It really does. If a clinician underestimates the cancer's aggressiveness and undertreats the patient, they're risking a lethal recurrence or distant metastasis. The cancer spreads before we can stop it.
Right, which is the nightmare scenario.
But the flip side is equally harrowing, honestly. If a clinician overtreats a patient who actually has a very stable prognosis, they're subjecting that person to aggressive systemic adjuvant therapies.
Like intense radiation or heavy chemotherapy regimens.
Exactly. Treatments that carry severe toxicities. We're talking about permanent damage to surrounding organs, early menopause, a drastically reduced quality of life.
Clinicians desperately need, like, a master diagnostician to help them perfectly tailor these treatments. And that brings us to the focus of today's show. Okay, let's unpack this. We're looking at a really fascinating paper published in the journal IET Systems Biology in 2026.
Yeah, this was led by Shen, Jiliang Yang, Fuquan Zhang, and their esteemed colleagues.
Right, and they didn't just go hunting for a single new biomarker. They built a complex, multimodal deep learning model. They essentially trained an AI to act as that master diagnostician by fusing together raw pathology images, standard clinical data, and complex genetics.
All to predict whether a patient will have short-term survival, meaning under 3 years, or long-term survival, which is over 3 years.
But before we get to the AI's actual predictive power, we have to look at the raw materials it was given. An algorithm is ultimately just a reflection of its training data.
It is. Garbage in, garbage out, as they say. The researchers utilized 2 completely separate data sets here. First, a training set of 119 cervical cancer patients pulled from the Cancer Genome Atlas.
Commonly known as TCGA, right?
Mm-hmm.
That's a massive, publicly available database researchers use all the time.
Exactly. But to validate their model, they didn't just, split that TCGA data in half. They manually collected an entirely independent testing cohort of 53 patients from Peking Union Medical College Hospital, or PUMCH.
I want to pause on that, actually, because anyone listening who works with machine learning knows that having a completely independent data set from a different physical location is the absolute gold standard.
Without a doubt.
Because if you only test your AI on data from the same hospital it trained on, the AI might just, memorize the quirks of that specific hospital.
Yeah, it happens all the time in medical AI. An algorithm might learn to recognize the specific digital artifact of the scanner used at Hospital A, or even the specific way Hospital A prepares their slides.
Rather than learning the actual biological markers of the tumor?
Precisely. By forcing the AI to prove itself on a completely independent cohort from a different hospital in a different city, you ensure the model has actually learned the biological truths of cervical cancer. It proves the model is generalizable.
So regarding the data itself, they fed the AI 3 distinct modalities. It's kind of like solving a crime. You don't just want the suspect's description. That's the clinical data, like age and tumor stage.
Right.
You want their DNA, which is the molecular data, specifically the somatic gene mutations. And you want the security footage, which in this case are the histopathological images. A standard H&E stained biopsy slides.
That's a great analogy.
But let me push back on this approach for a second. Staining a tissue sample with H&E hematoxylin and eosin, it's a very old-school physical and chemical process. You literally have lab techs dipping glass slides into vats of dye.
Very true.
One lab might leave a slide in the pink dye for 10 seconds longer, or the chemical batch might be a few days older.
Doesn't
Combining all this highly variable, messy visual data with precise genetic data just create a noisy, chaotic mess for the AI.
This raises an important question because it absolutely can. And this is the central challenge of multimodal machine learning. Routine batch effects in pathology labs are just a nightmare for algorithms.
I can imagine.
If you feed those natural staining variations into an AI, the algorithm might accidentally learn a false correlation. It might decide that dark purple slides mean higher mortality, simply because all the dark slides in the training set happen to come from a batch of patients with more aggressive cancers.
Right, it's learning the dye, not the disease. So how do they fix that?
To prevent this, the researchers had to be incredibly rigorous. They used an automated quality control tool called HistoQC.
How does HistoQC actually strip away that human error?
It mathematically analyzes the images before the AI ever even sees them. HistoQC looks at the color histograms, the brightness thresholds, and the contrast ratios of every single slide. Yeah. If a slide is washed out, blurry, or overstained, with eosin making it way too pink, the tool flags it and filters it out. It ensures that only pristine, structurally clear tissue images make it into the training pipeline.
Okay, so the data pipeline is squeaky clean. Let's look at what the data actually tells us. And I want to start with the genetics because, intuitively, DNA is the ultimate biological blueprint. It dictates everything a cell does.
It's the source code.
Right. So I naturally assume that the molecular data, the specific genetic mutations in the tumor, would be the absolute strongest predictor of whether a patient survives beyond 3 years.
Well, it's a logical assumption, and the researchers tested it thoroughly. They used a statistical method called Lasso-Cox regression analysis to sift through the genomic data.
Lasso, that stands for least absolute shrinkage and selection operator.
You got it. It's a fantastic mathematical tool for this kind of work because it actively penalizes complex models.
Meaning what exactly?
Meaning, if a gene has no real impact on survival, Lasso forces its mathematical coefficient all the way to 0. It essentially deletes it from the equation. It leaves you with only the most critical variables. Out of thousands of genetic data points, Lasso isolated 484 genes significantly associated with survival.
And out of those 484, 3 specific genes stood out like massive red flags. The genes RGR, DBN1, and CLLCR. The hazard ratios on these mutations were huge.
Let's define what that means in this context for our listeners. A hazard ratio measures how much a specific variable increases the risk of an event, in this case, mortality. A ratio of 1 means no effect.
Right.
These 3 genes had hazard ratios, indicating that if a patient's tumor carried these mutations, the risk of dying from the cancer spiked dramatically. And this aligns perfectly with existing biological literature. DBN1, for instance, produces a protein involved in remodeling the actin cytoskeleton of a cell.
So it affects how the cell physically moves and changes shape.
Precisely. And if a cancer cell can easily change shape and move, it can detach from the primary tumor, enter the bloodstream, and metastasize. We've seen DBN1 drive metastasis in colorectal cancer.
Wow.
Similarly, CLCR is a known driver of poor prognosis in acute myeloid leukemia, so the AI successfully identified highly dangerous genetic culprits.
Here's where it gets really interesting, though. I get super confused by the results of this paper right here. We have these powerful, validated genetic markers. But when the researchers tested the 3 modalities individually to see which one predicted survival the best, the molecular data performed the worst.
It did.
It achieved an area under the curve in AUC of just 0.559. And for context, an AUC measures the overall performance of a classification model. An AUC of 0.5 is a literal coin flip. 1.0 is perfect prediction.
Exactly.
So 0.559 is like barely better than random guessing. Did they just have corrupted sequencing data from the TCGA? Because if DNA is the literal blueprint of the cell, how does it fail to predict the outcome?
The sequencing data was perfectly fine. The flaw wasn't in the data. The flaw is in our assumption about how cancer operates in the human body. It comes down to the fundamental difference between genotype and phenotype.
Okay, walk me through that.
Genetics give us the genotype, the blueprint. They tell us what the tumor has the potential to do. But a blueprint doesn't build a house in a vacuum. The tumor exists in a complex neighborhood called the tumor microenvironment.
So the surrounding tissue is actively interfering with the blueprint.
Exactly. A cancer cell might have that terrifying DBN1 mutation screaming at the cell to move and metastasize, but what if the patient's immune system recognizes the threat and successfully walls off that tumor with dense, impenetrable connective tissue?
The cell wants to move, but physically can't.
Right. Or what if a secondary biological pathway in the host tissue alters the local chemistry, silencing that mutated gene so it never actually produces the dangerous proteins? The DNA tells you what the tumor wants to do, but the phenotype, the physical structural manifestation of the disease in the tissue, dictates what actually happens.
So if the genetic blueprint is lying to us, where's the truth hiding? It turns out we have to physically look at the battlefield to see who is winning. Which brings us to the whole slide images.
Yes. To analyze the tissue phenotype, the team deployed a convolutional neural network called ResNet-18 to look at the H&E stained images.
The whole slide images are massive files. We're talking gigapixels of data. You can't just feed an image that large into a neural network and expect it to process it.
No, it would crash instantly.
Right. So they broke these massive tissue landscapes down into tiny manageable patches, 512 by 512 pixels each. They discarded any patch that was more than 30% blank space, just empty glass. And then they applied a mathematical technique called Marchenko color normalization.
Marchenko normalization is a brilliant solution to the lab batch effects we discussed earlier. H&E staining relies on two dyes. Hematoxylin stains the cell nuclei a dark purple-blue, and eosin stains the cytoplasm and connective tissue pink.
Right, the classic pink and purple.
Marchenko normalization mathematically decomposes the image to separate the specific color vectors of the stain from the underlying optical density of the tissue.
It's essentially stripping away the Instagram filter of the specific lab, leaving only the raw architectural shapes of the cells.
That's a great way to put it. It ensures that ResNet-18 is judging the structural integrity of the cells, the size of the nuclei, and the density of the tissue, rather than the intensity of the lab technician's staining technique.
So ResNet-18 looks at thousands of these normalized, tiny image patches, and the results are stunning. Using just the H&E images alone, the AI achieved an AUC of 0.725.
Which is a massive jump.
It completely outperforms the clinical data, which scored a 0.647, and it makes the genetic mutation data look entirely useless. What's fascinating here is, what is the AI actually seeing in those pink and purple pixels that tells it if a patient will survive?
What's fascinating here is that it's seeing a hidden language in the tissue that human pathologists cannot consciously quantify. A human pathologist looks at a slide and grades a tumor by looking for cellular atypia. How weird the cells look, and counting mitosis, which is how fast the cells are dividing.
Right, standard grading.
But ResNet-18 is looking at complex pixel gradients. It's calculating the spatial relationships between tens of thousands of cells simultaneously across the entire slide. It's detecting subtle textural patterns in the connective tissue that correlate strongly with patient survival.
Okay, so the images are a great predictor. The clinical data is an okay predictor. The ultimate goal of this paper was multimodal fusion. How do we combine them to get an even better prediction?
To marry these vastly different data sets, the researchers used a sophisticated technique called multimodal compact bilinear fusion, or MCB.
Let's break down how MCB actually works, because it's not just adding the scores together. I like to think of it like judging a meal. The image data is like the visual intuition of a master chef. It's highly nuanced. The clinical data, like the patient's age, is like a strict nutritional label. It's a single hard number. MCB fusion doesn't just put the plate next to the label, it mathematically cross-references them.
It computes the outer product of the visual features and the clinical features, projecting them into a massive, high-dimensional mathematical space.
Right, so it's looking for hidden correlations. In the medical context, MCB takes the 128 high-level visual features extracted by ResNet-18 and mathematically multiplies them against the patient's age, discovering complex interactions between how the cellular architecture looks and how old the patient's immune system is.
And this fusion was highly successful. When they tested this combined image and age model on the completely independent PUMCH dataset, it achieved an AUC of 0.783. Combining the visual landscape of the tumor with the clinical context of the patient creates a remarkably robust predictive tool.
But wait, I have to point out a major contradiction in the data here.
Oh.
So if images plus age gives us an accuracy of 0.783, then throwing the genetic data back into that fusion must push the accuracy over 0.8, right? You're giving the AI the absolute full picture of the disease.
You would certainly think so. But the researchers tried that. They combined images, clinical data, and the genetic mutation data, and the accuracy actually dropped.
Wait.
Yeah, it fell to 0.686 on the training set. Adding more complex biological data made the AI worse at its job.
Why on earth does giving a supercomputer more information make it dumber?
This is a perfect example of a phenomenon in machine learning known as the curse of dimensionality, which is severely compounded when you have small sample sizes. Remember, the training cohort was only 119 patients. When you introduce genetic mutation profiles into the math, you're introducing massive variability.
Let me see if I can visualize this mathematical noise. Imagine trying to train an AI to predict if it's going to rain tomorrow. You feed it photographs of cloud formations, and you feed it the barometric pressure. The AI gets really good at predicting the rain.
Makes sense.
But then you decide to also feed it the serial numbers of every single umbrella sold in the city that day.
That is exactly what happens. The umbrella serial numbers are real data, but they're so hyper-specific and random across a small sample size that the AI gets confused. It starts chasing false patterns.
It's like, oh, a serial number starting with 7 means a thunderstorm.
Exactly. It stops looking at the clouds and starts learning the noise. The genetic data in this study contains so much individual variability that it introduced mathematical interference. The AI started focusing on rare mutations that didn't hold true prognostic value across the broader population.
So in machine learning, throwing more data at an algorithm isn't always better. You have to throw the right data at it.
Exactly. A universally applicable clinical feature like age, fused with rich tissue imaging, proved far more stable than noisy genomic variants.
That makes total sense.
Yeah.
Now, we have to address the massive elephant in the room regarding AI in healthcare. Medicine requires interpretability. An oncologist can't look a patient in the eye and say, we're going to put you through aggressive chemotherapy because a black box algorithm spat out a number.
No, doctors need to know why the AI made its prediction. Interpretability is the biggest hurdle for clinical adoption. To pry open the black box, the researchers utilize a technique called GradCam that stands for Gradient-Weighted Class Activation Mapping.
How does GradCam actually let us see into the AI's mind?
It works backward. Once the AI makes a prediction about a patient's survival, GradCam traces the mathematical weights backward through the layers of the neural network, all the way back to the original H&E image.
Oh, that's clever.
It calculates which specific pixels had the strongest mathematical influence on the final decision, and it generates a heat map over the tissue. Red areas indicate pixels the AI cared about. Blue areas indicate pixels it ignored.
And the heat maps generated in this study were incredibly revealing. For the patients predicted to have short-term survival, meaning they had highly aggressive, lethal cancers, the AI's heat maps lit up heavily in the stromal regions.
Right.
The stroma is the connective tissue that surrounds and supports the tumor. The AI actually paid less attention to the dense clusters of bulk tumor cells. But for long-term survival predictions, the heat maps flipped. The AI focused heavily on the actual tumor cells themselves.
If we connect this to the bigger picture, this observation bridges the gap between computer science and biology. To dig deeper into what exactly was happening in those stromal regions, the researchers took the images and ran them through a secondary neural network called HoverNet.
HoverNet.
Yeah, it's specifically designed to locate, segment, and classify individual cell nuclei. It examines every single cell and categorizes it. Is it a neoplastic tumor cell, an epithelial cell, connective tissue, an inflammatory cell, or a dead cell?
And when they cross-referenced the cell counts from HoverNet with the actual patient survival times, they discovered something profound. The density of inflammatory cells and dead cells in the tissue were highly correlated with patient survival.
And this correlation achieved a concordance index, or C-index, of 0.76.
A C-index is basically a grading system for survival models, right?
Yes. A score of 1.0 means the model perfectly ranked every patient's survival time in the exact correct order. A score of 0.5 is random chance. Achieving a 0.76 using just cell counts is an incredibly strong biological signal.
So when the AI predicted short-term survival by staring at the connective tissue, it was actually looking at the inflammatory battleground.
It was. For decades, the field of pathology focused almost entirely on the tumor cell itself. How big is the nucleus? How weirdly shaped is it? But this AI aligns perfectly with a massive paradigm shift in emerging oncology literature. The tumor microenvironment, specifically systemic inflammation and how the host's immune system reacts to the invasion, is a primary driver of cancer progression and metastasis.
The AI wasn't programmed in an oncology textbook. It didn't even know what an inflammatory cell was. It just looked at the pixels, looked at who survived, and deduced a profound biological truth entirely on its own.
It derived biology from pure mathematics.
The host's inflammatory response in the surrounding tissue dictates the lethality of the disease. That is incredible.
It's a remarkable validation of the model's clinical utility. It isn't just finding statistical noise, it's finding known critical biological mechanisms of cancer progression.
So what does this all mean? What's the ultimate takeaway for you, the trailblazers, listening to this? What this paper proves is that by fusing routine, inextensive H&E pathology images with incredibly simple clinical data like a patient's age, we can create highly accurate, reliable prediction models for cervical cancer survival.
We don't necessarily need to order $1,000 genomic sequencing panels for every single patient just to predict their prognosis.
Exactly. It puts a powerful tool directly into the hands of clinicians. If this algorithm can analyze a standard biopsy slide and confidently predict short-term survival, the oncologist knows they must immediately escalate adjuvant therapies.
Conversely, if the AI predicts long-term survival, the oncologist can confidently spare that patient the severe, life-altering toxicities of aggressive radiation or chemotherapy. It is the realization of accessible precision medicine.
But I want to leave you with a provocative thought to mull over. Toward the end of the paper, the authors humbly acknowledge the limitations of their study. They note the relatively small sample size, and they mention the need for broader genetic diversity in future training sets.
Standard scientific caveats.
Right. But then they drop a very subtle hint about where this technology is heading. They mention the emerging use of hyperspectral and multispectral imaging of cell autofluorescence.
Oh, that is fascinating.
Think about what we just discussed today. This AI achieved a 0.783 accuracy using standard H&E staining. That's a chemical dye technique that was literally invented over 100 years ago.
It's analyzing century-old analog technology.
Exactly. So, if a deep learning model can predict survival this accurately just by looking at the physical shapes left behind by pink and purple dye, what undiscovered biological secrets will the AI uncover when we start feeding it hyperspectral images? Images that capture dozens of invisible light frequencies, revealing the foundational chemical signatures and metabolic state of the living cells themselves.
That's a whole new frontier.
We are just scratching the surface of what the digital tissue landscape holds. Thank you, Trailblazers, for joining us on this deep dive into the source material. We started this conversation talking about the broken diagnostic X-ray machine of oncology. But as we've seen today, by giving AI the right lenses, the murky waters of cancer prognosis are finally starting to clear. Catch you on the next one.